Hello, welcome to 13 Universal, and we shall still be solving MCQ questions on A-level biology, uh, immunity series 3, and I am Shedro. So let's go to the question. All right, get ready with your pen and paper. Let's solve. Okay. Now, the first question here says a student, consider a student wrote down three statements about antibodies. Which statements are true? Okay, their structure depends on peptide bond, hydrogen bond. Okay, that's all about that. So, it didn't show the answer immediately. Okay, would not even see the options. Okay, but let's check. Let's check at. Okay, why A is the answer. So their structure depends on peptide, hydrogen, and disulfide bond. Is this correct? Now, peptide bond forms primary structure. Yes, antibodies have primary structure. Let me show you. Antibodies, of course, it has a Y shape like this. Yeah, this is a long chain or heavy chain. This is the long chain. Have two long chains. It has two short. This is a short chain. This is a short chain. All right, watch this. So this short chain polypeptide is joined to long chain disulfide bond. The same here, disulfide bond joins them. Now the disulfide bond also join the two long chains. Okay, and then of course you can see that this is antigen binding site. That's the variable region. This is the constant region. This is hinge point. All right, so their structure depends on peptide bond. Yes, because this there has four polypeptide chains, three long polypeptide chains, two short. Okay, and polypeptide will have primary structure. Primary structure contains peptide bond. Of course, you can see that they have tertiary, which is this particular shape and that. And of course, it has quaternary, more than one polypeptide chains joined together. Hydrogen bonds and disulfide bond help to support quaternary and tertiary structures. So number one is correct. Okay. Two says that there are protein molecules with both tertiary and quaternary. That's correct. Tertiary, they have antigen binding site. Okay, and they're made up of more than one polypeptide chain. That's quaternary. Tertiary is precise 3D shape. And then of course we have four polypeptide provides four antigen binding site. No, they have only two antigen binding site. This and this, this and this. That's why 3 is wrong. Okay, so we have seen the answer. The answer is A. Is not A, is 1 and 2, which is B. Which is B. This is wrong. This is 1 and 2. Okay. 3 is wrong. There are only two antigen binding sites. Okay, please take note. Now, newborn babies have natural passive immunity. What is the correct explanation for this? All right, pause this video and try it. What is correct explanation for this? Anyway, see, this is not permitted. Is permitted. What is correct explanation for this? Immunity. Now, this is newborn babies have natural passive immunity. Immunity is not inherited. Okay? Immunity is not inherited. Is that correct? Okay? But it is passed on from the mother to the baby. Is it inherited or not? Okay? Now, antibodies are broken down. Antibodies are broken down. Yes. Okay? Antibodies are broken down. Yes. Immunity is not inherited. Okay? What is the answer? Okay? Is immunity inherited or not? Of course, it's passed on from mom to. Okay? All right. It's broken down. Yes, the child's enzymes will break it down in the stomach. Remember that antibodies are proteins. Okay? So these are proteins that will be broken down, digested by the protease enzymes in the belly, stomach of the baby. So antibodies are broken down, yes, that's correct. So passed on from the mother, yes. All right, let's look at this. Now, which is correct description of different types of immunity? Pause the video and check it. Natural, active. Natural means passed on from the mother across the placenta and through breast milk. So antibodies, but this is natural. No, natural active. Okay, that one I said is natural passive. Okay, that's natural passive. Okay, yeah. But natural active is due to infection. All right. Now, antibodies cross placenta. No, this is passive. Memory cells are formed. Yeah, memory cells are formed in active. Yes. Injection of antibodies. No, this is passive. Injection of antigen. This is artificial. So this is only one that explains at natural active. Memory cells are formed. Okay. Natural passive. So we follow through. Antibodies cross placenta. Yes. So it is natural, of course, from the mom naturally. All right. And then passive because immune response does not take place. No memory cells form. That's why it is called passive. All active immunity have their own memory cells that make their own antibodies. Okay. Artificial yes, injection antigen. The artificial passive injection antibody. So B, all right, looks like the answer. That's the answer. The World Health Organization (WHO) sets a target to eradicate polio using vaccination by the year 2000. However, cases are still being reported in some part of the world after this date. What explains these new cases of polio? What explains them? Okay, so please, I want you to solve this question. What explains it? Pause the video and solve this. Okay. Now, number one says some parts of the countries are difficult to reach because of poor transport or what? Yeah, this is correct. One is there. So which one has one? So this automatically eliminates D. Okay, we are focused with A, B, and C because one is correct, and D doesn't have one. The second one says there is not enough research to develop more effective vaccines. Well, I think there is enough research. Okay, World Health Organization, they are working on it. Do you think so? Why haven't they eradicated it? Where is polio found? The question, where is polio? Polio is found mainly in less economically developed countries, countries that are poor. Okay, all right. You don't hear polio in more economically developed countries, like developed nations. Okay, yeah. So this may not be the case. Okay, but let's look at the records of vaccinated and unvaccinated people are incomplete. Yes, that is why. See what it says. However, some cases are still being reported in some part of it, so it's not yet completed. So these two could be the reason, right? That's C. Okay. Now comment us. I want to hear you. I want to hear you. Okay. Challenge answers. Okay. Let's teach us. Explain to us. Want to learn from you. Have questions? Ask. All right. Say what are the functions of plasma cells during an immune response? Okay, during an immune response. Plasma cells. If you know it, let's do it. Destroy cancer cells, differentiate. All right, that's it there. So what do you think is that? Tell us the answer. Chairman, tell us the answer. Okay, chair lady, tell us the answer. So what are the functions of plasma cells during an immune response? Does it destroy cancer cells? No. What destroys cancer cells? T helper cells. Okay. No, T killer. It binds and destroys it. Okay, bust it. T killer, not plasma cells. Of these plasma cells from B lymphocyte differentiate into memory cells. Nope, they don't differentiate, but they do secrete antibodies. Yeah, that's what they do well. Plasma cells of B lymphocytes secrete antibodies. Okay, so let's move on. All right. The next question says some of the facts about a pathogen P are that. These are facts, guys. This analysis. Please pause the video. Let's see what you have. Try this question. Okay. Pause it and just try this question. All right. Fry this fish for me. I'm hungry. Says it is transmitted in food. This is information. Okay. And what this looks like cholera. Okay. It lives in human intestine. This is people cholera. It has many genes coding for surface. Okay. It changes its surface antigen. Okay, that's what it said. Has many genes, changes it. Okay. It may or may not trigger an immune response. Okay. Yep. Which explains why T is difficult to develop an effective vaccine for P. Why it is difficult, all right, to develop effective vaccine for P. P can mutate to produce different antigens. Yeah, because it has many genes. P is a eukaryotic cell with many genes. All right. Yeah. But this must translate to antigens because it's antigens that trigger immune response, non-self antigen. Right? This is correct, but this is more suitable. Right? Because this, if it has different antigens, that means it's going to trigger different immune response. Okay? Because each specific antigen triggers a particular immune response. Now, this A looks correct. Okay? C says P is found in continental food. That's not the reason for. Right? People can be no. The only reason for to develop effective vaccine is this. A. Let's go. Which description is correct for B lymphocyte? Okay. Pause the video. What do you think? B lymphocyte processed in the thymus gland? Nope. It's T lymphocyte that's processed in the thymus gland. That's why they call T lymphocyte, processed in thymus, T for time. Okay. So A and B eliminated. Focus on C and D. Release antibodies immediately after. No, they don't release antibodies. B lymphocyte don't produce antibodies immediately after formation. Is when there's immune response. Okay. Act in the cell-mediated response? Nope. They do not act in cell-mediated immune response. It's only T helper and T killer. All right. Can act as they act in the cell-mediated response? Nope. Can act as antigen-presenting cells. B lymphocyte can act as antigen-presenting cells. Okay. So what do you think is the most suitable answer here? Antigen-presenting cells is a cell that present antigen. Okay, if it comes in contact with an antigen, if it's binding to an antigen, can it act as, all right, antigen-presenting cells? So it looks like that is the most suitable answer there. Okay, all right. Antigen-presenting cells are cells that show they display antigen on their surface. Okay, all right, that's the most suitable answer here. All right, so okay. Now, it is the following event occur when a phagocyte responds to the presence of a pathogen. So this is it. Let's see what you got. Solve this question, please. So if you want to engulf pathogen, phagocyte needs to happen. What's the first thing that takes place? Okay. So let's assume that this is our phagocytic cell. This is that bacteria with its pathogen. The first thing that when it attaches to it, the first thing that happens is not to engulf it. Right, starts with engulfing. So it folds inward to produce a vacuole. Now it's going to fold inward, okay, and then it will produce a phagocytic vacuole. Don't mind my diagram. So you're going to have something like this. This is it, okay. So this is a phagocytic vacuole containing the pathogen inside, right inside there. Okay, and then the rest is it. Okay, so it's going to digest. Of course, you know what happens there. Lysosomes will fuse with this and then release the enzymes. The enzymes will digest the bacteria. All right, the peptidase enzymes will digest the break the peptide bond, digest protein. Of course, lipase breaks down lipids. Lysozyme breaks down peptidoglycan, of course, and then nuclease breaks down nucleic acids. So the first thing that happens is endocytosis. Yes, endo, it has to engulf it. So where is 1? Do you know what I started with? 1 is A. That's the answer. The other thing falls in place. The next thing is vacuole formation. That's vacuole formation. 5. Enzyme digestion. Yes, that's the next thing that happens. We're going to fuse, and then so the answer is quite A. Which type of immunity occurs as a result of ingestion of antibodies by an infant through its mother's milk? Now this is coming from the mother, so it's natural. Okay, so natural, B or D. But antibody is not made by the baby, it's made by the mother, so it's passive. Okay, D is the answer. All right. Okay, during a primary immune response, the following events occur. Okay, I wanted to check out this. Tell us the answer. What do you think is the answer? They said, in which order will event occur? This is primary immune response. Okay, so what's the first thing that happens? It says some B lymphocyte form plasma. That's not the first thing. B lymphocyte with specific cell surface divide. No, that's not it. Okay, now there must be a stimulation, of course. Let's look at, you see, this is T helper cells secrete cytokines. T helper cells identify a specific antigen, so it undergoes clonal selection. So once you identify it, so 5 should be there, right? First thing, and then the next thing that releases cytokines, which stimulate. So T helper cells identifies and then releases cytokines to stimulate appropriate B lymphocyte. So this is this and this. Let's check the next one. B lymphocyte with specific cell surface receptors divide. Yeah, it's stimulated to divide. So that's 2. Specific antibodies is produced. No. So the next thing, once cytokines are released, they will stimulate B lymphocyte with specific cell surface receptors to divide repeatedly by mitosis. Okay. So it's going to be 542. Of course, 1, the B lymphocyte form plasma cells, and the plasma cells will make antibodies. Okay. So C is the answer. Calm down, look at this again, chat us. All right, did you enjoy today's class? Okay, let's know what you think. Don't forget, we have more other topics coming from other subjects. Okay, and tell us what you want us to do for you. Which topic do you want to ask us questions? We want to help you, we want to be there for you. Don't forget that this is schedule. All right, see you next class. That's series 4 immunity. Bye.